In this video, we're going to talk about some of the principles in the practice of light microscopy as well as electron microscopy. Both these different types of microscopes in essence work pretty much the same, where a illumination source passes through a specimen. The light is then magnified until it finally hits something that detects the light that's passed through the different lens systems. In the case of the light microscope, that would be the human eye. In the case of an electron microscope, it's some kind of screen or television that is actually able to view the electrons. Looking at the parts of the compound light microscope, we can see that the light source starts down here at the bottom, passes through a special lens which increases resolution. We recall from our formula, resolution equal to wavelength divided by the numerical aperture of the condenser plus the numerical aperture of the objective. The condenser lens is one of these lenses that increases the resolution. Ultimately, it's going to then pass through the specimen and through an objective lens, and the objective lens also contributes to the resolution. But the objective lens also begins to magnify the specimen. The condenser lens, no magnification. The objective lens does magnify until finally it passes through a second lens in the compound binocular microscope, the ocular lens, and the total magnification is going to be equal to the product of the ocular times the objective lens. Finally, the magnified light passes through the ocular lens and enters into the human eye. Now, with our compound light microscope, our 1000x lens, the very highest magnification that we can get from the compound light microscope, unfortunately is right at the limits of resolution for light microscopy. And if we were to use our highest magnification lens, which is actually a 100x lens, as the light passes through the specimen into the objective and then ultimately to the eye, we would look at the specimen and we would see not a very clear image. And that's because when the light passes through the specimen in the slide, some of the light escapes from the bottom of the lens. We can solve this dilemma by putting in a drop of immersion oil. We've all learned in lab how to apply the immersion oil, and what that does is it refracts, it bends the light a little bit as it comes through the glass slide in the specimen. More light is actually captured and enters into the objective. And when we observe this image under the microscope, we see that our number 5 has gone from a blurry to a very, very clear and crisp appearance. The magnification, looking here between the left and the right, is completely unchanged. However, we can clearly see that by the addition of the immersion oil, we've had a fantastic increase in the resolution of our image. We're only going to want to use immersion oil with our highest magnification of possible with the microscope, the 1000x total magnification, which is the 100x objective lens. The different types of light microscopy that we can use in the microbiology lab are summarized here on this slide. We'll look at a little bit closer examples of each of these. One of our learning goals after finishing up this section of the course is to be able to look at a particular image that we might see under the light microscope and to be able to recognize what type of microscopy it would represent. All of these basically work exactly the same way that we saw outlined in the cartoon on the previous slide, with an illumination source passing through a condenser lens, then the specimen, and then the two magnifying lenses before hitting the human eye. But what we do with each of these is play a little bit with the light path, the path of light through here. We may interrupt it slightly, and that's going to have some interesting optical tricks that it can play and ultimately increase the resolution. Straightforward light from the light source through the lenses is what's known as bright field microscopy. And in bright field microscopy, we'll see an extremely bright background with perhaps a dark specimen on that slide. Now, with standard bright field microscopy, because microorganisms tend to not have much in the way of contrast on their own, if we're looking at a living microorganism, it would probably not show up in this bright pink color. In fact, there might be extremely little to be able to see at all. We'll see in the next picture exactly how that works out. But by interfering with the light path, as we see right here, we've stuck this little opaque disc into the middle of the light path. It changes the light that ultimately reaches the human eye. The light that hits the human eye is actually light that has been bent around the edge of the microorganism. This is dark field microscopy, and in dark field microscopy, of course, you see a very dark background with a ghostly white halo of the microorganisms surrounding it. If we further adjust the light path by putting this diaphragm lens, or this diaphragm blocking material here in the light path, just part of the light hits the specimen, and areas of the microorganism, areas of the specimen that have subtle differences in density are going to bend the light more or less than light that's just passing through the slide itself. And so the phase contrast microscope, it's the same microscope. All we've done is interfere with the light that's passing from the light source to the condenser lens. We've just interfered just a little bit, but this is going to show very subtle differences in the density, internal structure of that microorganism. And then finally, Nomarski differential interference contrast microscopy is going to use polarized light instead of straight white light, and it's going to give you an extremely clear, almost three-dimensional appearance of the living microorganism. So here we see our bright field microscope image. We can see in here, well, not really actually a whole lot. I can sort of see a ghostly outline of this material, something like this. And there are some dots in here, but not really a huge amount of structure. We can tell this is bright field because there's a very light background, and the specimen really doesn't have a huge amount of detail inside of it. We flip the switch and stick that opaque disc down between the light source and the condenser lens, and we've now set up dark field microscopy. Dark field microscopy, a dark background, and we can now see pretty clearly the outline of the cell that's on here, a nice bright white line surrounding there, and these things that were kind of darkish gray before, very easy to see at this point. Dark field microscopy is particularly useful for looking at living cells that might be moving, and white things would be very easy to spot. The movement of cells on a dark background would be extremely easy to be able to see. Dark field's main use is that it's fast, and you're able to spot movement pretty easily with it. Phase contrast microscopy, in this we've simply adjusted the light path a little bit more by sticking that annular phase ring between the light source and the condenser lens. Now, with phase contrast microscopy, the periphery of this microorganism is extremely easy to see. And in fact, we can now spot this organelle that wasn't apparent before. This is in fact the nucleus. The cell that we have here is actually a human cheek cell, and we can see these gray things that we could see before. In dark field they were bright white, and in bright field they were kind of lightish gray. We can see these things actually appear to be sitting on top of this cell, and these are actually bacteria that are sitting on top of this human cheek cell, viewed by phase contrast. How can we distinguish phase contrast from bright field? Well, number one, the background is going to be darker, but of course not black like in dark field, and we see a fair amount of internal structure. Highly visible with phase contrast. What's the use of phase contrast? Well, much like dark field microscopy, movement is going to be extremely easy to see. As we see microorganisms moving here, when there's a lot of contrast afforded by phase contrast, that movement is much easier to see. Additionally, if we have an organism such as this human cheek cell, we can begin to see internal structures that weren't apparent from before. Well, finally, Nomarski optics. The microscopes that we have in Bio 230 are not capable of doing Nomarski differential interference contrast microscopy. What separates a microscope that's able to do this type of imaging? This microscope costs about $2,000, and the microscopes that we have in Bio 230 can do phase contrast, they can do dark field, they can do bright field. York College does in fact have several Nomarski microscopes. They are really nice microscopes, but they are going to start at about $12,000 instead of the $2,000 that we saw before. With this, it's the exact same slide we were looking at before. The boundary of this cell is extremely sharp focus. These bacteria that we saw before are obviously laying on the top surface of this cheek cell. The last kind of microscopy we'd like to talk about is fluorescence microscopy. That's also not a kind of microscopy that the microscopes that we have in Bio 230 will be able to accomplish, but it has a lot of utility within the clinical setting, so we'll speak of it so that we at least understand the basis of it. Over here on the left, we see a prepared specimen. Not a lot of detail, kind of a clumpy, darkish material without really very much interesting for us to be able to see here. This has been specially treated with a chemical, and the chemical reacts with certain microorganisms that are in this big clump of material. And when we switch over to fluorescence microscopy, the chemical reacting with the bacteria in here begins to fluoresce or glow. It glows when we use ultraviolet light. The ultraviolet light hits the microorganism, hits the chemicals on the microorganism, and visible light is emitted. That we then see through the oculars of the microscope. This does give us a slight increase in resolution, slightly better resolution. Recall our formula for resolution, where resolution is equal to the wavelength divided by the numerical aperture of the condenser and the numerical aperture of the objective lens. And we recall that by playing with wavelength, we can lower wavelength to a point where we get better resolution, a smaller number for resolution. When we drop the wavelength down into the ultraviolet light, we'll have a much more improved resolution. But recall, ultraviolet light not visible to the human eye. Because in a fluorescence microscope visible light is emitted, we're going to get that increase of resolution that we wouldn't be able to take advantage of by lowering the wavelength to something not visible with the human eye. How are we going to be able to differentiate fluorescence microscopy, such as we see here, from dark field microscopy? Again, we're going to have a dark background, but the specimens here are going to be colored because of the chemicals. If we flip back to dark field microscopy, we can see that it's a dark background but white things within the field of view. Phase contrast or fluorescence microscopy, dark background on the slide, and colored specimens. We can see this applied within a clinical setting right here. The microorganism in question is coated with these chemicals that have a fluorescent dye attached to them. As ultraviolet light hits this fluorescent dye, a color appears in the dark background here, and this is a clinical diagnostic test to detect the presence of a microorganism, Treponema pallidum, which is the causative agent of syphilis. The best extension of visible light microscopy is confocal fluorescence microscopy. Its big difference from the pictures in the previous slide is instead of just using any old ultraviolet light, it uses a high energy laser to illuminate an extremely thin part of the whole specimen. It still uses fluorescent dyes to add contrast, but because of the use of this high energy laser, it gives the best possible resolution that we can get with any sort of a light microscope. Sometimes the images generated by confocal microscopy are fantastically beautiful. This is a picture of some human cells called HeLa cells In the act of mitosis, we can see the nuclei of these cells dividing in the mitotic apparatus right there in the center of it. Fantastic, really beautiful picture, really at the very limits of what we can see using the light microscope. Now, of course, some things that we might want to look at of microbiological interest are smaller than that limit of resolution of the light microscope. Remember, the limit of resolution of the light microscope is approximately 200 nanometers in size. So anything of smaller than 200 nanometers is not going to be something that's going to be visible by the light microscope. Fortunately, the electron microscope is able to visualize these things. Instead of using a light beam, a light source of visible light, it uses an electron beam to pass through that sample. The basic principle of the transmission light microscope and transmission electron microscope and the light microscope is exactly the same. My light microscope diagram here is now upside down from what we saw before. The light source is up here at the top, passing through the condenser, the specimen, the objective, and ocular lenses until it's seen by the eye. The electron microscope works basically the same, but it uses an electron beam that passes through a condenser lens, a specimen, an objective lens, and finally a final magnifying lens until it's seen on a television screen. So the path of the illuminating beam is pretty much exactly analogous to the light microscopes that we're already familiar with. Now, electrons are not visible to the human eye, so consequently, if you were to stick your eyeball down here, you wouldn't see anything at all, and furthermore, you'd be blind after you did that. And so we have to use a kind of a fluorescent screen, a television screen, in order to be able to view the pattern of electrons as they pass through this. Now, we'll recall that this limit of resolution of the light microscope was because of the formula: resolution, wavelength, numerical aperture plus numerical aperture. We said that the lower limit for this wavelength was basically ultraviolet light. Anything smaller than ultraviolet light was not visible to the human eye, so consequently we couldn't lower this number anymore because of the properties of visible light. You might guess from this, because we have a much smaller number r here, that the wavelength of a beam of electrons is much smaller than the wavelength of a light beam of visible light. Here we can see somebody actually working at an electron microscope. You can see it's quite a large piece of equipment. The electron gun sits up here, and this is what this power cord's plugging into. The specimen is right in here. It's stuck in through a little port there. There are some magnifying lenses, a condenser lens, objective lenses. Now, instead of using glass lenses, they use magnets. Magnets are able to bend beams of electrons, and the beams of electrons are bent and focused as they pass through the specimen until finally they hit the little fluorescent screen down here, and this person will use these little eyepieces in here to view the fluorescent screen that's down there. Here's a picture that I actually took when I was doing research before coming to York College. These are images of yeast cells. There's two yeast cells, a yeast cell up here, a yeast cell down here. This is the cell wall of the yeast cell. These are probably things like ribosomes, these little dots inside here. We can see a size marker up here. We indicate that this thing has been magnified 40,000 times its normal size. Transmission electron microscopy is viewing things through the specimen, just the way the light microscope does. I like to keep in mind this letter T that starts out the word transmission. If you see transmission electron microscopy, the light's passing through the specimen. To give you an idea of the small amount of detail that we can see here, these little black dots are 10 nanometer gold particles. You can clearly see these 10 nanometer gold particles. Blow this picture up onto onto this projection screen in the classroom. We can really see the really fine detail that we can get from the electron microscope. Here are some more pictures of transmission electron microscope pictures. Various sorts of organelles. This looks like something like a mitochondria viewed from the inside. Here we see a section of muscle, some kind of a cell, and another kind of a cell. In each case, transmission EM is going to let you see a cross section, the inside of a specimen. This is where its great power comes from. The scanning electron microscope is a little bit different beast. It's again going to use an electron beam, an electron gun, and pass that electron beam through some magnifying lenses, which again are going to be magnets, until finally it hits the specimen. But in the case of the scanning electron microscope, the electron beam bounces off of the surface of the specimen, and the reflected electrons are then collected by a collection device until it's finally viewed. And so in the case of the scanning electron microscope, instead of seeing the cross section, the inside of a microorganism, as we see over here on the left, we see the surface of that microorganism. I like to remember this: transmission through, letter T, scanning, surface. And so again, we'll just see a very incredibly fine detail of the surface of that microorganism. Here are some pictures of scanning EM images. We can see a bunch of different microorganisms, but in each case we're only seeing the surface of that microorganism, and we see no inside detail. A couple of words about the preparation of materials for electron microscopy. We recall that when we were talking about different kinds of light microscopy, we could play with the light source and set up dark field or phase contrast, Nomarski optics, and that would allow us to be able to view living specimens with the light microscope. One of the unfortunate drawbacks of the electron microscope is that no living tissue can be recovered from the electron microscope. First, electron beams are going to be extremely damaging to biological material, and so if we want to recover the material in an intact state after looking at it under the microscope, the electron microscope is not your choice. Let's just look here. This is the sort of procedure that is necessary for preparing things to get these really fantastic images that we see here and here. We start off, first the specimen has to be treated with a pretty nasty, harsh set of chemicals which cause the biological material to be stuck in one place. Things like formaldehyde and embalming fluid are used to fix these specimens. Next, that biological specimen is put into some molten plastic, which is allowed to solidify. And in the case of transmission electron microscopy, where you want to see the inside, you have to cut those cells into very, very, very thin pieces with an extremely sharp knife. And then finally, because biological materials don't tend to stop electrons very well, you then need to cover that specimen with heavy metals that is able to reflect electrons. The ones that are typically used in microscopy, electron microscopy, are going to be heavy metals such as gold or uranium. This table is going to summarize the different kinds of microscopy that we have available to us in the Bio 230 laboratory and also in clinical labs everywhere. We'll want to be very comfortable with the basic principles of each of these different kinds of microscopy. We want to be able to look at the different kinds of images such we've seen pictures of over the past couple of minutes and be able to recognize how a bright field light microscope image is going to look different from a dark field or a phase contrast, and how we could tell the difference between a transmission EM image and a scanning EM image.